protein synthesis happens in three main steps: transcription, mRNA processing, and translation. But we're just going to focus on transcription and mRNA processing.
Transcription has three main processes: initiation, elongation, and termination.
Transcription needs three things: DNA, RNA polymerase, and RNA nucleotides.
RNA polymerase binds to the promoter region of DNA that regularly includes a sequence called the Tata box. The RNA then moves down the DNA, transcribing a single strand of RNA.
As the RNA polymerase moves down the DNA from the 5' to the 3' end, the DNA is unzipped and one strand is used as a template. RNA nucleotides are assembled according to the complementary nucleotides on the template DNA strand. Although this process seems similar to DNA replication, there are a few key differences you must make note of. Only RNA nucleotides are used to make the mRNA. Only one strand of DNA is transcribed, and primers are not required.
Transcription ends or is terminated when the RNA polymerase reaches the stop codon, then detaches.
After that, the mRNA is ready to be processed.
Next comes mRNA processing. Before mRNA can be translated, it undergoes the following transformations. A modified version of guanine is added to the 5' end soon after transcription has begun. This cap is known as the 5' cap.
Transcription proceeds until termination, then the RNA polymerase releases the pre-mRNA. A poly-A polymerase binds to the strand, cuts off the 3' end, and attaches a sequence of 200 or more adenine nucleotides called a poly-A tail.
This tail helps the mRNA leave the nucleus, protects the mRNA from degradation by hydraulic enzymes, and helps ribosomes attach to the 5' end when it is in the cytoplasm.
After both ends have been modified, intervening sequences called introns are removed through a process called RNA splicing. Small nuclear ribonucleoproteins, or spliceosomes, splice the coding sequences, exons, from the introns, and remove the introns to degrade in the nucleoplasm.
The last concept is alternative RNA splicing. You must keep in mind that not all introns are non-coding. For example, one DNA strand transcribed into pre-mRNA could code for many different proteins, but certain introns are spliced out to make a processed mRNA code for a single protein. Sublamo.